Good morning, ladies and gentlemen. I'm not quite sure if the sound system is working, but I hope you can hear.
So this is a particle that somehow is involved in making proteins. Now, very soon after people analyze, the first thing you do with a particle is what biochemists like to do is simply destroy it and break it down into its components and figure out what the components are. Then they try to put it all back together again, and they did both of those for the ribosome. And what they found is it's about 2/3 RNA by mass, but has about 50 different proteins. And these proteins and RNA somehow assemble in a very specific way to form these large and small subunits of the ribosome, which together make up the entire ribosome. Now, how does it work? Well, that too involved a lot of work, and this slide is going to summarize about 40 years of biochemistry. So what it does is the ribosome latches on to the genetic message, and it has slots for those adapter molecules, the tRNA molecules. And it figures out how to start at the first codon, where the first amino acid has to be brought in. And the first tRNA then comes in and brings in that first amino acid. Then what happens is a new tRNA comes in. Now, of course, lots of tRNAs come in and go into the ribosome. If they're the wrong tRNA, which is to say they don't match up with this codon, then they leave the ribosome. They're sort of rejected. If they're the correct tRNA, the ribosome accepts it and joins up the first and second amino acid. And then the ribosome has to move, and then it gets to the next codon, and then of course the third tRNA comes in, and if it's the right one, it's accepted, and the second and third amino acid are joined. Now look what's happening during the process. One part of the ribosome is reading the code and only allowing the right tRNAs to add to the growing protein chain. So the amino acids are added in exactly the right order that is specified by the code. And at the other end, it has a pocket which stitches together these proteins, these amino acids, to build the new protein. And this carries on until it reaches the end of the message, and then it knows there's a special signal telling it when to stop. And that's how every protein is made. So you can think of this as a large molecular machine, because it's reading information, it's moving and making things, and it's at the same time using energy. So it's very much has all the properties of what we think of as a machine. Now, this is about the state that the field was in. We knew a lot about what the ribosome was, what it did. What we didn't know was what it looked like and how it did these things. And that's when I entered the field. Now, my entering the field was something of an accident. Okay, so I'll tell you how that happened. So I grew up in sort of Western India. I'm actually a South Indian, so I was a foreigner effectively from the age of 3, because India is a little bit like the EU. It's a collection of people with different languages, etc. But in a sort of common framework. And so then we. At the age of 19, I had a degree in physics, and I went off to America to get a PhD in physics. And I went to Ohio University, where I spent the first 5 years of my life in the US. Now, it was the only university that would give me a fellowship, and I was happy to take it and go there. But once I got there, I realized there was a problem. So the problem was this. I had no trouble with my coursework in physics. I did fine in all my courses, but as soon as I started research, I realized there was a real problem with physics, and that is the problems were very hard. Okay, and this by this I don't mean exam questions. What I mean is it's hard, really hard to do something original in physics, because physics is quite a mature field, and to do make fundamental discoveries in physics is, I think, still quite hard. And in fact, there have been very few fundamental breakthroughs. There have been a lot of what I would call applied breakthroughs in the previous few decades. And so I thought, what would my life be like? I'd started doing theoretical physics, and I thought, what would my life be like if I continued doing physics? And this is what I thought my life would be like. I would end up doing, spending the rest of my life doing boring calculations that I would keep publishing, you know, in Phys Rev B or something, but it wouldn't actually add to any understanding. And I thought that wasn't a great way to spend my life. And so at the same time, I was reading Scientific American. And, you know, remember, this was the 70s. You know, people had just figured out how to sequence DNA. The first structures of, you know, biological molecules were coming out. We were understanding cell membranes, all these breakthroughs in immunology, you know. We're understanding, you know, acetylcholine and its effect and so on. You know Jean-Pierre Changeau here, etc. So, you know, it was a tremendously exciting field. And I have to say, the revolution in biology is still going on, okay? And so every issue had a major breakthrough. And I knew that many physicists had made this transition before. So I thought, well, maybe, you know, I should really start over and try and become a biologist. So I applied to a bunch of graduate schools. Many of them wouldn't even look at me. They said, you're already going to have a PhD, so we can't accept you to do another PhD, you know. And one of them, Yale, said, okay, we can't accept you into a graduate program, but maybe somebody will offer you a postdoc. And I got 2 offers, one from Don Engelman, but one from Tom Steitz, which is odd because I ended up sharing the Nobel Prize with him. But I turned them both down because I didn't know any biology. I said, look, you know, I'm not ready to do a postdoc. So instead, I decided to go to graduate school again, and there were 3 universities that accepted me for graduate school again, even though I already would have a PhD. And one of them was UC San Diego. So my son was about 6 weeks old, and my stepdaughter was about 7 years old, and I packed up all our belongings in a Ryder truck and drove all the way from Ohio to San Diego, where I spent 2 years here at the University of California in San Diego. And the first week was a kind of, you know, introductory survey lectures for first-year graduate students given by professors about their work. And I realized I was hopelessly lost. You know, biology, if you don't know anything about it, it's a complete alphabet soup. They're talking about, you know, genes which have, you know, 3-letter acronyms and, you know, things like that. If you don't know the vocabulary, you have no idea what they're talking about, okay? So I realized I had to take undergraduate courses. So for the first year, I took, you know, basic biochemistry, cell biology, and genetics, you know, just to learn what a graduate student would normally know coming in. And at the same time, I did lab rotations, and there's some very comical episodes described in the book about what it's like if you don't know anything and you go into a biochemistry lab and want to deal with C14 glucose. Okay, so anyway, after the first year, I realized I knew quite a lot of biology. I'd caught up, I'd done lab work, I knew how to work in a lab, I knew how to run gels and columns and so on. And so I realized maybe I didn't need a second PhD. So I read another Scientific American article, and one of the authors was Don Engelman, who had offered me a postdoc. And I wrote to him and I said, look, you offered me a postdoc when I didn't know a damn thing about biology, but now I actually know something and I know how to do work in a lab, and so maybe you'll be interested now, 2 years later. And he said, sure, why don't you talk to my colleague Peter Moore and maybe we'll set you up. So 2 years later, we ended up moving to Yale, where I ended up working for Peter Moore, who was Jim Watson's student. You know, Watson's lab also pioneered a lot of ribosome research. And so I went to his lab, and his lab was working on using a physical technique called neutron scattering to look at where all the 20 or so proteins were in the small subunit of the ribosome. Now, remember, we didn't know what the small subunit looked like. We didn't even know what these proteins looked like. We just knew roughly where the center of mass of each protein was in the ribosome, okay? Doesn't seem that interesting now in hindsight, but at the time, nobody had any idea of how to tackle such a large, complex as the ribosome. So I thought any information would be interesting. And also, when this work was started, the whole RNA world hypothesis had not been established yet. This is before Czech and Altman discovered that RNA could do catalysis and that early life might have evolved from RNA. So there was this feeling that proteins did a lot of the various functions of the ribosomes. At least it would be useful to know where they were. Okay. Now, after this, I couldn't find a single university job. I applied for 50 university jobs and got 0 interviews. I ended up spending a disastrous year in Oak Ridge, and then I ended up at Brookhaven National Lab, where I started doing work on the ribosome. Now, the problem was I found myself at a dead end, a sort of second dead end. My first dead end was physics. My second dead end was I was using a rather useless technique to look at biological structure, okay? And so what is the problem? The problem is, if you want to understand a molecule, you have to know what it looks like. In fact, that was true right from sodium chloride. You know, chemists thought of sodium chloride as a fixed diatom, okay? And it was Bragg who showed that it's not true. You know, sodium chloride is sodium and chlorine ions, you know, all separated and forming a checkerboard in a crystal. And that's been true for proteins and so on. And the idea is the following. If you want to know how a car looks like, you know, if you're hovering above the Earth like some Martian looking at cars, you'll just see them as little dots moving around. And you won't know much about what a car looks like. And if you get closer, you know roughly its shape, and you know it has wheels and rolls on wheels. That's not going to tell you that much. But then you start opening it up, and you realize there's a steering wheel, there's an engine, somehow they're connected. You're starting to learn more. But then when you look at a car in real detail and know how every component is connected and what the function of each component is, that's when you start learning and understanding how a car actually works, okay? And it's the same with molecules. And so the question is, how would you visualize a molecule? And normally when you want to look at a molecule, normally when you want to look at a small object, you would use a magnifying glass or a microscope, and you would look at it. And what a magnifying lens does is it collects scattered rays from the object and recombines them in order to make a magnified image. But you have to remember that the lens is not adding information that isn't there. So the information is there in the scattered rays, and whether the lens is there or not. All the lens is doing is recombining them to form a magnified image. Now, it turns out there's a law in physics that you can't resolve objects that are closer together than half the wavelength of light that you're interested in. And of course, I know that now people have circumvented that by using some tricks. But the tricks are actually, they're not actually looking at the object, they're looking at fluorescence from the object, and they're able to control it. So it's not as if you can just take any object and look at it and avoid this physical limitation. So how do you then look at molecules? Because molecules, the atoms in a molecule are about 1,000th the distance of the wavelength of light that you're going to use. So the way to do that is a technique called crystallography, where you coax your molecules into forming crystals, and then you hit them with X-rays. And what you then do is the X-rays will scatter, but unlike a normal, just any old object, which can scatter light in all directions. For example, the light from the screen goes everywhere in the room. It doesn't matter where you're sitting, you should still be able to see the screen. But if you have a crystal, it turns out that because of interference from this regular array, it reinforces the scattering along certain directions. So the scattered rays look like an array of spots like this. And so you would then measure the scattered rays, and then what you would do is, instead of doing what a lens does by recombining them, we can't do that with X-rays. What you do is you simply measure the scattered rays, and in a computer you do what a lens actually does. So you recombine the information in a computer, And so you're effectively doing what a lens does, which is performing a Fourier transform on the scattered rays. Now, I didn't know any crystallography, and I thought, what's the best way to learn crystallography? I thought if I stayed where I was, I would just be fussing around and not focusing. And I thought the best thing to do is to go away on sabbatical. So I wrote to Aaron Klug, who was the director of the MRC lab of molecular biology in Cambridge. And of course, I was a fairly unknown scientist, and he was a Nobel laureate who would then go on to become president of the Royal Society and the director of one of the most famous labs in this field. And luckily for me, he didn't throw away my letter into the waste paper basket, but he replied and said he was interested, and he sponsored me for a Guggenheim Fellowship. So I went there for a year. I learned crystallography. Then I collaborated with a colleague of mine in Brookhaven. And what we initially did was to take each of these proteins and crystallize them and solve their structure. So this is what the detailed structures look like. But of course, we were looking at their structure out of context. This would be like if you want to understand how a car works, instead of looking at a car, you'll go to an automobile parts store, okay? And you'll say, I want to look at a spark plug. I want to look at a gas pipe or steering wheel. And you have no idea how they all fit together, but you're looking at each component, and saying, oh, this is what a spark plug looks like, without any idea what it does or where it actually sits in a car, okay? So it was okay for a while, but then again, it was another kind of dead end. Now, the real path that eventually led to the sort of resolution of this problem was started when Ada Yonat, an Israeli scientist, wrote to Heinz Gunther Wittmann, who was a director of the Max Planck Institute for Genetics in Berlin, and asked him if she could come and spend some time in her lab. Now, Wittmann, as I point out in the book, had always been interested in trying to do the structure of the whole ribosome. And actually, the first person he hired to do it turned out to be a complete fraud, and he was exposed in an article in Nature. His name was Hasko Pardies, and then he shortly afterwards left his position. And then, but in the end, he ended up being a faculty member in Britain, and so. Well, anyway, so that was one thing, and then another person was Bob Frederick, who's a well-known crystallographer at UCSF, who wanted to go work with Wittmann because he had a German girlfriend, but it turned out she dumped him, and so he was no longer interested in going to Berlin. And but then Wittmann had this fellowship, so when Ada Yonat wrote to him, he simply transferred the Humboldt Fellowship to Ada, and then so that enabled Ada to go and work there. And after a year of doing some other stuff, she thought it might be a good idea to give whole ribosomes a crack. And of course, Wittmann was already interested in that, so he was very happy to support her. And the two of them then started this collaboration, and it resulted in crystals of the large subunit of the ribosome. And then a few years later, Maria Garber's group in Russia, and this is Maria Garber, they started working on a different species, and they were able to crystallize both the small subunit and the entire ribosome. Okay, and this character here, who looks like a Cossack, is Marat Yusupov. He still looks a bit like that, and he works in Strasbourg. So and he's quite well known, very well known in our field for his subsequent work. So these crystals have been around. So the first crystals were 1980, subsequent crystals were the mid-80s. And so they'd been around for quite a while in the mid-1990s by the time that I had become frustrated with solving little pieces of the ribosome. And the state of play was that 15 years after the first crystals, there's still no progress towards an actual structure, not even a low-resolution structure. Now, the first crystals were not good enough quality to get an atomic structure, but after some real persistence, Ada Yonat was able to get good crystals of the 50s. But then, many years later, there was still no structure, not even low-resolution structure. So the Yale group in the mid-90s of Tom Steitz and Peter Moore decided that maybe it's time for other people to start attacking this problem. And they decided to use Ada Yonat's crystals to see if they could make progress with it by trying their own ideas for how to solve it. Now, I didn't want to do that, and I thought, let these two fight it out for the 50S, because I didn't want to be a third person working on the same crystals. And I thought, well, let me, there are no good crystals of the small subunit or the whole ribosome. And I thought, well, the whole ribosome is a little too hard. I didn't think that I could get good crystals of it very easily. It had been around for quite a long time and not improved. But I had some ideas for how to improve the crystals of the small subunit. And I thought it would also be, it was about half the size of the large subunit and might be easier to tackle. And I thought once I tackled that, then maybe we could go on to something like the whole ribosome. So I was at the University of Utah at the time, and the small subunit is only small in comparison, relative to the large, but in fact it was enormous. It has something like 150,000 or 200,000 atoms. Okay, so it's not really trivial to even consider. It's about 1 million Daltons in molecular weight. So I persuaded two first-year graduate students, Bill Clemens and John McCutcheon, that this is a good idea for a PhD project. People have been working on it for 15 years with large groups and have not made any progress, but I think it's a good idea. Now, neither of them knew anything about either ribosomes or crystallography, so they said, yeah, it's a great idea. And so they started working on it. And this is a lesson, actually. If you know too much, you know all the reasons why it won't work. Okay, and so sometimes it's not such a good idea to know too much. And the reason I say that is this is Brian Wimberly. He was a terrific scientist. He was a postdoc, knew a lot about structure and RNA. And at the beginning, he really didn't want to join this project. Okay, in the end, he turned out to be essential for solving it. But in the early stages, he was too reluctant because he knew all the, how risky it was. Now, if that wasn't bad enough, I thought I would have plenty of time. Since the other two were focusing on the 50S, I could quietly work on this for a while and see how I got. But I went to a meeting soon after we had started working on this in Sweden. And, there I met Ada Yonat with her close associate, Francois Franceschi, and this is Steve White. And this is, as you can see from this picture and the last one, I was in my Bin Laden period. So the fact is that at this meeting, the Yale group showed that they were making progress on the 50S. And Ada showed that she thought that the 50S was not worth tackling. She thought there were problems with it. But she showed that that the 30S crystals that she had that didn't diffract so well, she had somehow found a condition by adding heavy atoms to them that stabilized them so they now were of improved quality.